Okay, so as Bilur sort of gave a good introduction, I feel, for what I'm about to talk to you about, I'm going to basically present our work on network-based stratification, and this is work that was done here in UCSD in the lab of Trey Eidecker, where I've recently graduated and I'm now postdocing for the time being. So basically, just still a very brief introduction. Cancer therapy is still much in this regime of giving a standard of care to most patients coming into the clinic, and basically hoping for the best. And it's very often tailored for whatever is the common version of the tumor. Of course, we all, I feel, in this room share the sentiment that the way forward is to basically tailor treatment to specific patients groups or even specific patients, and basically think of how to, discover the right groups and match them with the right treatment. And this is not a new idea. It's been around for a long time. This is work from Todd Golub, at the time in Eric Lander's lab, where they basically used gene expression to divide a cohort of AML and ALL patients into distinct types. And this has been a subject of much interest in the subsequent decade. This is work from 2010 from the TCGA pilot project, where Verhaak et al. Basically defined 4 subtypes of glioblastoma that you could see here. And one of the nice results that they could demonstrate is basically this difference in survival patterns for certain subtypes. And so we were very curious whether it would be possible to do this instead of an expression in somatic mutations. And so somatic mutations, in case anybody here doesn't know, is when you, take profiles from both the patient's tumor and the patient's blood, compare the two using genome sequencing, and look at the difference, basically. So we're only looking at somatic mutations as opposed to germline mutations. And of course, you should start by, if you want to discover subtypes, we should start with something off the shelf. And we tried what we believe is one of the better clustering methods out there. So here we apply NMF and we basically get this result, which is totally nonsensical. To understand the problem or the reason we feel this is the case is basically the result of 2 forces. One of them is this sparsity and heterogeneity of cancer, which results in what has been often described as the long tail. Here we sort of see the histograms of the rows and columns. Here I just show a sample of chromosome 17, and you could basically see, if you were to look here, that most mutations occur very, very rarely, right? And this is characteristic of most cancer types. Ovarian cancer is no different. And of course, many of us in this room, and I feel many clinicians also in the cancer community are coming to realize that cancer is a disease of functions that has common functions among different cells. Another way to say this is that it has specific hallmarks that need to be disrupted. And another way to think about it is that there's basically some underlying network that describes the way that cancer genes basically cause tumorigenesis, right? So this is a nice illustration showing prominent cancer mutations, cancer-causing mutations in the form of a subway map. We were curious how would we use this to basically inform on subtype discovery, and we published this method nearly a year ago in Nature Methods. And basically, the main idea that I will describe next, and some of our more recent follow-ups, is that we take different networks, and so we took 3 networks, Pathway, Common, String, and Humanet. And all of these are what I sometimes like to jokingly call everything in the kitchen sink networks, right? Because they basically have every sort of edge you can imagine, right? They have literature co-citation, right? They have co-expression edges. And basically, everything is aggregated together. One piece of advice is when you're using these methods to only use the very highest confidence edges, which is what we do in this work. And so I'll quickly go over the method. And so the first step, and I think a step that in our hands has been is a rather powerful step, is this idea of moving from features that are in distinct genes to features on a network, right? And we've called this network smoothing. It's also been referred to as network propagation. And Bilor basically used a very similar technique in her work that she just presented. Very, very briefly, the idea is that you basically use some linear algebra to go from a discrete vector, although it does not have to be discrete. So here we have genotype A in yellow and genotype B in blue, right? And they are discrete and sparse. And we use this technique to basically see what is the network region around each one of these mutations, right? And what you see is that there are regions that sort of become highlighted. And now, instead of the sparse vectors we had before, we sort of have dense vectors that have a lot more elements to compare. And so we basically, the next step is we apply network clustering. And so we take those propagated profiles, we apply an NMF that we've modified to also include a regularization term for the network. And basically, we repeat this in the context of a consensus clustering algorithm. So finally, we basically get a patient-by-patient co-occurrence matrix, co-clustering matrix that we could basically report subtypes for. And really, when you compare these two, they look nothing alike, right? Which is something that we found quite profound and interesting. And we can show this in simulation. Here, we sort of compare driver mutation frequency in a simulated framework to the size of the cancer-causing pathway. And what we basically see here, anything that has colors is basically a meaningful clustering. And we could see that a lot more of the space is red when you compare these two without the smoothing and with the smoothing. And if you go to a random network, which I think is really an essential step when you're doing network-based approaches, you could see that a lot of this signal, the majority of the signal is again lost. And of course, I wouldn't be belaboring you with this, possibly some of you for the second time, if this weren't to work on real data. And so here we basically compare the results against standard consensus clustering to this network-based approach, and we could see the difference is quite striking. When you move these features from their original gene-based space to a network space, the results are profound. And so we tried this with the 3 different networks. We wanted to see this is not a network-specific effect of some kind. And we get similar clusters. I will focus on the results we felt are most interesting in the human network. So here we basically look at patient survival in the context of ovarian cancer. And really, you could see that we find 4 subtypes of different sizes. Or another way to think about this is that NBS is basically peeling off some subtypes. One of them has really different survival outcome. And just crossing the significance threshold, we basically tested for association with cisplatin response. So this is a proxy for cisplatin response, which is the time to disease recurrence. And basically, it's really striking that this subtype that seems to be the sickest is also not responding well to cisplatin. And the reason we think this is very interesting is that cisplatin is still the standard of care for ovarian cancer, right? So a woman coming into the clinic with ovarian cancer, that would basically be the treatment that she'd get if if the oncologist thinks she can tolerate that sort of chemotherapy. And so and many of these patients just sort of don't seem to respond to that therapy well at all. And so one of the things that is obviously interesting, right? So you found some subtypes. What do we do with the next patient that comes into the clinic? And so one of the ways to sort of leverage our results is to now train a classifier, right? That basically goes from our subtypes based on a genotype, right? And the classifier, really, the way to think about it is some function that goes from an input matrix to a result matrix. And we, in this case, used a boosting tree-based approach, but there's nothing unique to this function, right? You could think of many different functions that could do this. And one of the things, when we published this paper, we had there was only the TCGA data available, so there wasn't really a cohort to replicate no independent cohorts. And since then, the ICGC put together an Australian cohort of ovarian cancers of approximately 90 patients, and I was really curious whether this is going to pan out or not, and I was actually quite fortunate that it did, and it would what I found to be surprisingly little finagling. There's this thing where you're going to replicate your data, it's from a different group, from a different lab, right, and you spend hours or days or weeks, right, trying to get the data in a way that your results replicate. So this is really, on my first pass, we got this result, which we find quite striking. 3 of the 4 subtypes are recapitulated. The 4th subtype possibly being too similar to subtype 3, or just having too few training examples is not reproducible. But again, we find a striking similarity in the overall survival trends. And we were also pleasantly surprised to discover that the cisplatin response that we were finding is again recapitulated, and it's totally independent cohort from a different continent using different technology. This was done on whole genome sequencing as opposed to the exome sequencing and the TCGA, really done years apart, and this classifier is able to pick up this difference. We were really curious whether we could also show this with cell lines, which is never trivial. And actually, if you take the classifier and you apply it to the CCLE, you find, so this is, to be perfectly honest, sampling from the extremes. The classifier is actually not as good on the CCLE data, but when we sample from the extremes, and we could take different thresholds of what we define as the extremes, you see a difference in terms of the response to cisplatin in the direction that we would expect, right? So patients in subtype 1 are more resistant to cisplatin response, which again we found quite interesting, and we're currently following up on this story. So the last couple of slides are unpublished work that we're trying to do functional analysis and basically understand better what is going on in this type. So switching to something. A different, newer example to illustrate the power of this method is this work from the TCGA research network if somebody here, the people here saw the talk by Ilya the other day, he mentions extensively. So this is a really nice TCGA work where basically we looked at thyroid cancer. And one of the things that are characteristic of thyroid cancer is that the mutation frequency is actually much lower than ovarian, lung, and endometrial that we've looked at before in our original publication. And so we were really curious, could we basically aggregate other types of genomic perturbations into this framework and use them to basically have more coverage of the space. And so we basically aggregated these three types of features that were kindly provided by the collaborators in this team effort. We included replicated gene fusion events and basically the logistics in homozygous CNVs. And we basically treat them as part of this same framework. And again, we managed to find very interesting subtypes. We focus on this 3 subtypes result. And one of the main findings from the TCGA paper is this distinct difference between BRAF and RAS-like phenotypes in thyroid cancer. Giovanni Cirello from MSKCC basically defined an expression-based biomarker that predicts whether you're more RAS-like or more BRAF-like. And one of the things we were really pleasantly surprised is to see that our 3 subtypes really track this result nearly perfectly. We basically find 3 subtypes where the middle subtype is this intermediate that actually tracks this RET fusion subtype. And so this could also be shown to track other forms of expression biomarkers, and particularly the differentiation score is quite interesting, where you see this, again, RET fusion that I think this is all mentioned in that paper, but it's not, we actually don't dig any deeper into this, is this intermediate between these 2 expression phenotypes. And you could also connect this to clinical phenotypes. These are all obviously very correlated, but I'd like to point out, for example, here this risk category, which shows that the patients in subtype 3 basically exhibit a much higher chance of recurrence. And one of the nice things that we could do now is we could basically go into each one of these subtypes and ask what is the network, or what are the set of features, right, that distinguishes that type. And here we basically use the hotnet method from Ben Raphael, who are also co-authors in this paper. And we basically together come up with these, this is here a network for the BRAF subtype and a network for the RAS subtype. And here we could see a network for the RET subtype, which is quite interesting, right? We sort of see these different RET fusions, and there are also a couple of NTRK3 fusions that are sort of characteristic of this subtype, and some of the other mutations that sort of tend to co-occur in patients of this type. And so what I've shown you here, or what I'd like to believe I've shown, is that network-based stratification recovers biologically relevant subtypes of cancer. Subtypes can be recapitulated using a classifier independent cohort, and subtypes appear to have specific affected subnetwork. One thing that I sort of don't have time to show exactly is that these affected subnetworks are actually distinct from the global subnetworks that are found for these types, right? So if you were to do this network-based analysis on the entire cohort, you get completely different results, which is actually quite sensible if we believe the hypotheses that these are, in fact, distinct cancers just occurring in the same tissue. So this is a one-slide summary. We basically go from something that really has very little information to something that we'd like to argue has a lot more information simply by doing a rather simple network transformation. So basically by looking at these features through a network perspective, and these track clinical phenotypes in a meaningful way. I'd like to acknowledge some fellow contributors from the lab. So JP Shen is an oncologist postdocing in our lab and who really helped in this work. Hannah Carter, who I think just left, used to be a postdoc in the lab, is now starting her own lab and is actively recruiting postdocs. Andy Gross, a fellow grad student, Justin Wang, and of course Trey Eidecker for all his work and help in this project. We have some other collaborators from UCSD. Thank you all for listening, and I'll be happy to answer any questions.
Hi. So superb work. Your network processing approach is very general, right? It has little to do with cancer. So my question is this sort of something that we should all be using when trying to interpret networks?
So I think it's actually like one of the things that you point out quite well is that this is an extremely naive approach on some level, right? Like we basically just, instead of looking at specific features in the terms of genes, we now move to a different feature space. I think that's a nice way to think about it. That is basically activity on a network. And it seems to work, and it seems to even reproduce, right? So I think the short of it is yes. We should all be trying to think of, particularly in cases where we don't have a reason to believe that, different cancer mutations, we already know, can result in identical phenotypes, right? They're basically different parts of the cell that are being perturbed, right? And causing the same functional result, right? And the network that we've used here are basically a proxy for getting at that, right? Seeing that these are similar functional regions of the cellular network that are being hit.
So is there something special about the network in which you applied this method, or should it be generally applicable?
I think it's fairly generally applicable. These are, very standard networks that we've used, and we've seen this work in 3 different networks. I've seen this work in other networks too, not to the same extent, right? So we started this project with more networks, and I don't think there's any specific network. The one thing that I can say and that we're actively exploring is are there specific types of networks that work better for specific types of problems, right? And I don't really have a chance to show you.
Thank you.
Great talk, Matan. I was going to follow on actually that line of reasoning. When we do pan-cancer analysis, we like to relate, a patient who has ovarian cancer with somebody who has endometrial sometimes if they look like they're molecularly similar because we might be able to borrow treatments. And so, and then we've seen a lot of talks here that say, depending on the tissue, the network's different. Right? There's plenty of talks like that were very exciting. So I wanted to hear you speculate on using these network-based signatures in a pan-cancer setting, but where the networks might be different. Are you thinking about along those lines? I mean, how can it be generally usable in that sense?
We're certainly thinking along those lines, and we've actively pursuing a pan-cancer version of this work. There are many challenges to that, as I'm sure you know from coordinating that effort for the TCGA. I think it's a great idea and something that we may have not explored enough. There's definitely, the question is, we sort of lack an experimental technique that really gets us context-specific network, right? We sort of have naive approaches. One suggestion that there was a talk this week about using expression data, right? There's all sorts of challenges about using expression to basically get at what is the tissue-specific network. I very much hold the belief that this is sort of like a housekeeping network that I'm using right now, and I think there's a lot of work to be done to get at tissue-specific networks. I mean,
So you showed that one of your subtypes correspond to people who weren't responsive to chemotherapy, and I was wondering if you actually look at the genes in that network for that subtype and the genes in the other networks, do you see that there's genes that are supposed to be targeted by the chemotherapeutic drugs that you're not seeing in that subtype, or if that's not the case, are there genes that are common in the other subtypes that aren't in that subnetwork that might suggest some possible unexpected effect of the chemotherapy?
we do, and you could look at some of the things we point out in the paper. We didn't get many of the obvious things, like the one obvious pathway that seems to be preferentially hit in this subtype is the FGFR pathway that has been shown before to be related to cisplatin response, specifically in ovarian and other cancers, which we think is very interesting, and that's one direction we're following up on. Another category that was sort of very much enriched is this category of cytoskeleton genes, which there are some hypotheses. It's more like almost like nucleoskeletal genes. There are some hypotheses about how that could sort of serve as a mechanism for cisplatin resistance. But again, like this, we're still pursuing the functional part of this work, and we think it's a very interesting. And this is work that's being led right now in the lab by JP Shen, that I've mentioned in my acknowledgements.
Thank you.